Welcome to an edition of CCSD Living Environment. Today we're going to talk about cell transport. Cell transport involves how do cells get things in or out of them. You may remember that the cell membrane, which we just finished talking about, the job of the cell membrane is to kind of control what can move in or out of a cell. It's selectively or semi-permeable. Remember, that's primarily based on size. Well, for us to talk about cell transport, we need to look at this idea of a concentration gradient. Gradient means difference, the difference between. Concentration is the amount of. So it's the difference between the amounts of something on two sides of a cell. Well, the definition that you need to write down, the difference between the concentration of a particular molecule in one area and its concentration in an adjacent area. Adjacent means the area next to. So if you have something here, the adjacent area would be right next to it. So they give this very simple picture of a hill. The top being a high concentration, the bottom being a low concentration. If you start at the top and you need to ride your bike down the hill, it doesn't require you any energy. You can just coast down. If you start at the bottom and you need to go to the top, that does require you quite a bit of energy because you've got to pedal and invest time and effort to get there. So if we need to go from low concentration and push everything to a high concentration, that requires energy from the cell. Essentially, we're moving uphill. Another way to look at that is if you are supposed to be cleaning your bedroom. Well, if we talk about all the stuff that's on the floor and all the stuff that's in the closet, chances are, because you've been asked to clean your bedroom before, the closet is stuffed so full, it's the highest concentration it could ever hold. And so when you try to cram more stuff in there, low to high, requires a lot of energy. You have to have 5 or 6 friends over to help you close that door, instead of just putting it all away. Your mother would be happy to hear that in the video. So the idea of a concentration gradient is just the difference between the amounts of whatever molecule on two sides of a cell membrane. So we're going to talk about that idea of passive transport. Requires no energy. Remember, energy is ATP. So when we're undergoing passive transport, if something is passive, it's not really doing anything. It's passive. It's just kind of there. No energy. So passive transport requires no ATP from the cell. And there's 3 different types of passive transport that we need to look at. The first one is called diffusion. Diffusion is by far the most common example of passive transport. Diffusion is the random movement of molecules from an area of high concentration, where there's a lot of it, to an area of low concentration, where there's a little bit of it. Or we could simply say we're going down or with the concentration gradient. We're going from high to low. The bike is going down the hill. So I've got my high concentration, 2, 4, 6, 8, 10, 12, 14 molecules inside that cell, to my low concentration where there's only 3. Very simply, those molecules just move out. Very easily done. Diffusion will stop when there's an even distribution of the molecules, once it's reached equilibrium. And in this case, we truly mean equilibrium or balance, not dynamic equilibrium. It's definitely at a balance. So however many there are inside will match however many there are outside. It moves until it reaches a balance. And that's as far as it'll go. That is diffusion. And I believe in your note packet, right underneath that, it gives you a mnemonic device, hopefully that will help you remember. Make no confusion, it must be diffusion. Which way does it go? From high to low. There's a couple things that affect how diffusion will work. We can actually speed up how quickly diffusion takes place. The first one is with temperature. If we make it a little bit warmer, the molecules tend to move faster, so diffusion will take place faster. And the other one is size. The smaller the molecules are, the faster they're going to diffuse. And you know, if we're dealing with a cell membrane, that size really is what determines what will move in or out of that cell membrane. If it's too big, it doesn't make any difference how warm we make it. It's not going to go through there. So the smaller molecules will diffuse faster, and there is a point where the molecules are just too big. They'll never diffuse. So our second type of passive transport we call facilitated diffusion. If you facilitate something, what does that mean? Well, if you facilitate it, you help it out. You kind of help the process happen. So with facilitated diffusion, we have carrier or channel proteins in the cell membrane that increase the rate that some molecules cross the membrane, like large molecules or charged molecules that we call ions. Those proteins in the cell membrane are specific to the substance they are carrying. So if we have a larger molecule that we need to get into the cell membrane, these proteins are there to help give it a channel or to help carry it in. So I've attempted, with my artistic skills, which we all know are somewhat questionable, I've attempted to give you an example. Here's my cell membrane, I've got my phospholipid bilayer, and I'm exampling here two proteins. This one would be a carrier protein. Specifically, we call those channel proteins, because you can see there's a channel in between the two sides of that protein that something like an ion, a charged particle, would be able to go right through. In this case, we just call them a carrier protein. What's going to happen is a molecule will come from up above, it will come into the top of this, it'll go to the space, the bottom will open and allow that molecule to move into the cell. The molecule wouldn't be able to pass through the phospholipid bilayer on its own. So what it needs is to have this carrier that's going to grab it from one side and send it to the other. It's carrying it, if you will, through the cell membrane. Here, the protein is just creating a channel so that the particle can run through the cell membrane because it can't go through the phospholipid bilayer on its own. With facilitated diffusion, probably one of the most important things for you to remember, it is a type of passive transport, meaning that there's still no energy. We're going from high concentration to low concentration. Yes, there's a helper, but it's still diffusion. There's no energy. We're moving from high to low. And finally, the last type of passive transport that we need to look at is called osmosis. Now, chances are most of us have heard of osmosis before. Osmosis, by definition, is the diffusion of water into or out of the cell. So in this case, it's water that's moving from high to low. So the diffusion of water is called osmosis. Water moves from solutions with higher water concentration to solutions with lower water concentration. So pictorially, I'm trying to represent it here with my little dots. The blue ones are my water molecules. The orange one are some other molecule. So in this case here, there are more water molecules than there are other molecules. So we would say that this has a higher water concentration. In this case, there are more orange molecules or other molecules than there are water molecules. So we would say that this has a low water concentration. So which way is the water going to go? It's going to go from high to low. If these two containers were connected and they had a membrane between them similar to your cell membrane, it would allow the water to move from the high concentration to the low concentration. We're not concerned about the orange particles. The only thing we're worried about with osmosis is the movement of water. And water will always go from high to low. It's always going to go from where there's a lot to where there's a little. And an example that we have in living systems is with a plant cell. This picture is also in your note packet. Don't worry about drawing it. So if I look at my first cell, it's a plant cell. It's got its nice rectangular shape with a cell wall. And then you can see the cell membrane is crammed right tight to the cell wall, and the stuff in the middle, as we've talked about before, is the cytoplasm. So if I were to take salt or sugar water, a concentration of salt or sugar water, and I surrounded the plant cell with that salt or sugar water, what you would see happen is the water would move out of the cell. And if the water moves out of the cell, that's going to cause your cell membrane to shrink. So my cell wall stays the same, but the cell membrane shrinks, and you can see the cytoplasm, which is primarily water, has kind of squished in. The cell has shrunk. Again, where is there a higher concentration of regular water? Inside the cell. Salt water outside, so which way does the water go? From high to low. So the water moves out, causing my cell to shrink. So I can reverse that process. If I turn around and I put the cell into distilled water, Now there's a higher concentration of regular water outside the cell, so that water is going to move into the cell. So if that water moves into the cell, that's going to fill my cell membrane back up, and my cell's going to expand. So again, my cell wall hasn't moved much, but my cell membrane has come back out almost to the place it was when we started the whole thing. It's about the water moving. The water moves from high concentration to low concentration. Higher concentration inside, so the water moved out. Higher concentration outside, so the water moved in. There's a couple vocabulary words that we need to throw out there so we can stop using highly scientific words like stuff and thing. So, solution is a uniform mixture of two substances. For example, the salt water or the sugar water that I was talking about. If you were to take and create a sugar water solution, you mix the sugar into the water, and that's what you have, is sugar water, like Kool-Aid. You have water, you've got the powder, you mix them together, now you have Kool-Aid to drink. That's a solution. They're pretty much evenly mixed. The solvent is the dissolving substance, like water. Water is the universal solvent. If we're going to dissolve something, we usually dissolve it in water. And then we have a solute, which is the dissolved substance that you're putting in the water, like the salt, the sugar, the Kool-Aid powder, however you want to look at that. So instead of saying the stuff in the water, we're going to say the solute in the water. And the water then becomes the solvent. That's what we dissolved it in. And together they make up a solution. So based on those words and using those words, we need to look at the effects of osmosis. How does water move? The first thing that we want to talk about is something called a hypertonic solution. What does hyper mean? If you've got a hyper little kid, they've got a lot of energy, maybe more energy than we think they should have. So they're hyper. Well, a hypertonic solution, the concentration of solutes, or stuff, outside the cell is higher than the concentration inside the cell. So the cell is going to shrink. Again, remember, it's about the water moving. So where is there more water? There's more regular water inside the cell, so the water is going to move out of the cell, causing the cell to shrink. So the water moves out with a hypertonic solution. Yes, there's more stuff outside, but where is there more water? There's more water inside. What you have to remember is it's about the water. The water is what's moving. The next example we have is with a hypotonic solution. And I'm sure by now you've figured out hyper means more, so hypo means less. With a hypotonic solution, the concentration of solutes, or stuff, is lower outside the cell than inside the cell. So the cell is going to blow up. In other words, what am I saying? There's more water outside. So which way does the water go? From high to low. So all that water is going to move in and the cell's going to blow up. Not necessarily what we want to have happen. We need to be careful, especially if somebody's going into a hospital to be hooked to an IV, that we're not using, say, distilled water. So all that water rushes into the cells and blows them all up. So they need to take some of those things into account, and that would be a good life example of where this stuff plays out. And finally, the last one is an isotonic solution. Iso, if you remember an isosceles triangle, two equal sides, isotonic solution, the concentration of solutes is equal inside and outside of the cell. So water will move in and water will move out, but there's no net gain either way. It's just equal. Water moves in, water moves out. It doesn't matter too much. This is what we want if you do have to go in and get an IV in the hospital. You want an isotonic solution. You don't want it either way. You don't want it so your cells are going to shrink. You don't want it so your cells are going to burst. We want it isotonic. Let them fill up till they reach where they should be, and then just kind of hold tight in that place. Again, I'm going to say it. Remember, it is all about the water. It's about the water, it's about the water, it's about the water. It's not about the stuff. The stuff isn't moving. The water is moving. The water is what's moving, not the stuff. The water moves from high concentration to low concentration always. Always, always, always. So the question you'll be asked is, how come when you go to the movie theater they make your popcorn salty? Well, because the salt dehydrates my cells. No, don't give that answer. The answer to that question is because there's more water inside your cells than there is in the popcorn, so the water moves out of your cells to the popcorn, shrinking the cells in your mouth, and that's what makes you thirsty. It's not about the salt. You could do that with socks. If you put socks in your mouth, please don't try that at home. If you put socks in your mouth, there's going to be more water in your cells than in the socks. So the water is going to move out of the cells of your mouth and into the socks, and the cells are going to shrink, dehydrating the cells. Saved by the bell, I guess, kids. That's the end of our video for today. There is a part 2 to this video. Please make sure you watch part 2 so you get the rest of cell transport. Thanks so much, and I'll see you later.